In this video, we will discuss about mechanism behind TGF-beta induced motility. In the previous video, we have understood that the beta-4 region of the SMAT3 pathways plays a role towards cell motility in TGF-beta induced conditions. So here they need to find out the signaling pathways that get disrupted or that get triggered when the cells are induced with TGF-beta. From the literatures, they are able to find out that it's the GTP-RAC1 activations that will cause the Lampodia formations, which is a phenomenon that occurs when the cells migrate or move from one place to another place. So, and they wanted to check this GTP-RAC1 activations when the TGF-beta is induced with the cells. So they did this experiment in the wild type A549 cells, and they are able to find out that after 12 hours of TGF-beta induced, they are able to see these GTP-RAC1 activations. Then they check this activated GTP-RAC1 in the A549 SMAT3 knockout, and also when this SMAT3 knockout cell lines infected with this mutated SMAT3 and the wild type SMAT3, and they are able to see that in the case where the mutated SMAT3 has been infected, they are not able to see upregulations of the GTP-RAC1, but in the wild type SMAT3 they are able to see upregulations of the GTP-RAC1. So indicating that the A549 SMAT3 knockout, which is infected with this mutated SMAT3, will not be able to trigger this Lampodia path mechanism that helps during the in the cell motility. And then in the literatures, from the literature, they are able to see that there is Lampodia formations or GTP-RAC1 activations is also been GTP-RAC1 is also been activated using the P13K pathways. So what it did is they did these experiments in the wild type A549 cells with P13K inhibitors, and they are able to see a decrease in the migrations or the cell motility in the case where they use P13K inhibitors. So in the compared to the normal case where TGF-beta induced cells shows proliferations and migrations, but in the case where P13K inhibitors are used, they see a decrease in the proliferations and migrations, and these things are quantified on the right hand side, where we can see that when no P13K inhibitors are used, the migrations is statistically significant compared to the case where no TGF-beta is induced in the cells. But when the P13K inhibitors are used, they can see that the migrations is non-significant whether the cells are induced with TGF-beta or not. And thereafter they check these GTP-RAC1 activations levels in these P13K inhibitors, and they are able to see that the GTP-RAC1 is unable to activate in this when the P13K pathways are being downregulated, indicating that the GTP-RAC1 is being activated by the P13K pathways that get triggered by the SMAT3 pathways somehow. And when this SMAT3 pathways is mutated, they are not able to trigger P13K pathways, and thereby the GTP-RAC1 pathways is not been activated, and then Lampodia mechanisms for which is required for cell motility is not been triggered in those cell lines. Now, so these experiments in the penalty is done on the A549 cell lines. They did the similar experiments on the SMAT3 knockout cell lines which are infected with the mutated SMAT3, and in that case they wanted to check whether the SMAT3 pathways get disrupted in these mutated cell lines or not. And they can see that in the TGF-beta induced conditions, they can see the phosphorylated SMAT2 at 1 hour, but it is downregulated after 4 hours. But when they use P13K pathways, they can see a continuous rise in the phosphorylated SMAT2 pathways, indicating that the P13K pathways inhibitors or this A549 SMAT3 knockout cell lines, when they are infected with this mutated SMAT3 and the P13K pathways, they can see a SMAT3 signaling pathways are being activated, but these P13K pathways are not being activated. And thereafter they check the literatures and find out that it's the ROCK inhibitors. Some of the ROCK inhibitors pathways might downregulate these GTP activations. So they wanted to check whether it's the rock pathways that downregulate the GTP-RAC1 activations in these mutated SMAD3 infected cell lines or not. But they see a quite reverse result, that when they use the mutated uh, SMAD3 infected A549 cell lines, they are able to see the signals for the GTP-RAC1 when these rock pathway inhibitors are used. And similarly, they are able to see the GTP activations in the A549 SMAD3 knockout cell lines with ROCK inhibitors and without ROCK inhibitors. So this indicates that in the mutated SMAD3 infected cell lines, there is a GTP-RAC1 inactivations get triggered, but when these ROCK inhibitors pathways are been ROCK inhibitors are being used in these mutated SMAD3 A549 infected cell lines, they are able to see activations of the GTP-RAC1. This concludes that it's in the case of these mutated SMAD3 pathways, there is a de-repressions of this GTP-RAC1 when these ROCK inhibitors are used, and some of these GTP-RAC1s are being inactivated in the case of the SMAD3 knockout cell lines. That prevents the lymphodia formations that finally leads to the low cell motility. So I hope you understood to some extent these mechanisms behind TGF-β induced motility. If you need to understand more about these experiments and their conclusions in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.